I thank the organizers for inviting me to this meeting. All of my work is focused on understanding various mechanistic underpinnings of how the immune system functions, and then trying to harness that knowledge toward practical ends. To work toward this goal, over the last 15 years, we have tried to bring together approaches that have traditionally been stovepiped in different disciplines. In particular, we've tried to bring together theory and computation that's rooted in physics and engineering with the diversity of different biological experiments and human clinical data. A conceptual framework that we use a lot in our work is statistical mechanics. Its central goal is to understand how fluctuating, interacting microscopic objects can conspire together so that coherent mesoscopic and macroscopic behavior can emerge. So given that many of the processes that underlie the immune response involve cooperative dynamic events with many participating components, and many of these processes are stochastic in character, perhaps it's not surprising that bringing together these seemingly disparate disciplines of statistical mechanics and immunology has been somewhat fruitful. Now, these experiments in clinical data that can test predictions emerging from the theory and computation have been done in collaboration with various experimental and clinical immunologists over the years, most of whom are at medical schools. And with the key assistance of these collaborators, indeed my teachers in this field, over the years we've had some luck in shedding light on a variety of different questions in basic immunology and in human disease. But none of the work that I've done before has ever pertained to inflammation of the innate immune response. So I truly thought that this was an error, that there was some email mistake that they said this to me. But nonetheless, I had a phone conversation with people, and they assured me that it was not an error, and that it would be sufficient and appropriate if I described to you some of the methods that have been fruitful in adaptive immunity, and perhaps during the discussions we can see whether some of those could also be useful in thinking about innate immune responses and inflammation. And if that's not so, I hope I can still keep you entertained over the last 45 minutes or so. I'm going to speak today about work that pertains to HIV and human disease. But let me say a few words about work on this scale, pertinent to the molecular mechanism of T-cell signaling or cell signaling in general, that I think could be quite useful in innate immunity, especially if you were to make models that were one step deeper to tie in with the kinds of models that were described in the first two talks. So the kinds of things that people have done in the past in this field in adaptive immunity are, first of all, just cell signaling mediated by cytokines or receptor engagement. And there are a variety of different methods that people have used. Stochastic methods, such as master equations and rule-based simulation tools, which can be very useful, and also ordinary differential equations and partial differential equations, such as were described in the earlier two talks. Another very important class of methodologies that are being developed and have been developed that could be useful are sort of data-driven models where you can construct network models of gene transcription and RNA-seq data. In fact, in Yoram's talk, there was reference to one such paper on Th17 Treg differentiation that was led by Aviv Regev in the computational space. And her work is just one example. As you also know today, in characterizing populations of cells, instead of doing FACS, people can now do mass cytometry, where you can simultaneously measure more than 50 now different markers in single cells at the same time. This CyTOF instrumentation, which has been greatly popularized and developed by Gary Nolan's lab, yields data which is something like this. You see many, many markers. These do not need to be markers of innate immunity. They could be of all parts of the immune system. And then each cell, what is the value of that marker? So you can see this is a very, very high dimensional data set. And so if you think about how many two-dimensional analogs of FACS plots you would make, this would be an astronomical number. So how do you think about this? Well, the simplest way you can think about it is perhaps by doing principal component analysis. So you can cluster cells into different discrete phenotypes. But we all know that these cellular systems do not have an underlying basis which is linear, which is what the assumption is for principal component analysis. So how do you go beyond that? So there's a lovely little method that was developed by computer scientists some years ago, but in fact by Vandermaat and Hinton, which is called nonlinear dimensionality reduction through stochastic neighbor embedding. And basically what it tries to do is construct one two-dimensional representation of this high-dimensional data, which keeps cells that are close together in the high-dimensional space also close together in this underlying two-dimensional space. And we have employed that method, and Gary Nolan's lab and us together, but independently, have developed very similar methods to do that. Ours is called Accents. And what it does, it takes this very, very high-dimensional data, and ultimately it will automatically cluster cells in one two-dimensional plot, which is not a linear PCA, and you will see different cellular populations emerge. And for example, work done in collaboration with Mark Davis's lab, we have been able to identify a new type of CD8 T cell phenotype because of the presence of this high-dimensional data. And I think similar methods could be useful in thinking about single-cell data on innate immune systems as well. Well, now with that bit, I'm going to go into a more detailed description of a certain class of problems to show you how we have tried with the various computational methods to address some challenges in one field. So it'll be more of a drill-down talk for the rest of the period. And the topic I will concern myself with is a scourge on the planet. My frequent visits to Africa over the last 6 years have made vivid the havoc that HIV/AIDS is wreaking in that part of the world. So for example, I visit this area around Durban in South Africa. It's called the KwaZulu-Natal province. And in this province, about 60% of the women in their 20s are HIV positive. And when you combine that with the poverty that you see in those areas, it becomes very clear that a vaccine is urgently needed, and antiretroviral therapy is not sufficient for this population. So as has been demonstrated many times, a major barrier to the quest for a vaccine against HIV is the high mutability of the virus. So in these phylogenetic trees, the number of N's corresponds to the diversity of circulating strains. And you can see that the diversity of the flu virus in the whole world in a particular year is comparable to the diversity of HIV strains in a single infected individual, and completely dwarfed by the diversity of strains in just one region of Africa. So this high mutability presents a major challenge for vaccine development, because HIV can mutate to evade vaccine-induced antibodies and T cells that are specific for a certain strain. This is also the reason why there is no known example of HIV being cleared by natural immune responses, because even if you direct an immune response that could be problematic for it, then it can mutate to evade within the host. So really, design of a vaccine against HIV is a truly hard problem, because you are tasked to design strategies that will induce potent immune responses that do not seem to arise naturally, or if they do, very rarely. So that brings up two questions. What are such potent immune responses, and how do you induce them? So in the antibody world, partial answers to this first question are known, because many people, some of whom work with Tom back there, and some work with us like Dennis Burton, have identified antibodies extracted from patients that at least in vitro seems to be able to neutralize a diverse variety of HIV strains. They're called broadly neutralizing antibodies. And the epitopes that they target are more or less known. But still, how to induce these broadly neutralizing antibodies by vaccination is not known. In the T cell world, what's a good T cell response against HIV is beginning to emerge, but how you raise it by vaccination is certainly not known. So we work on both these questions. I'm going to describe our work in this space second, and begin with addressing the second question, or as a metaphor, how to hit HIV where it hurts with T cells. So because of the high mutability of the virus, there has been interest in focusing a vaccine-induced immune response to target single residues of HIV proteins that appear relatively conserved when you look at sequences of HIV proteins derived from virus samples extracted from diverse patients. The idea, of course, is that if a T cell targets such a residue, then HIV would have to evolve a mutation there to escape the T cell response, but that should likely make the virus unfit, because why else do you see that particular residue relatively conserved? But this strategy has been of very limited efficacy if you look at humans or monkeys that naturally target such residues, because HIV can evolve other mutations, so-called compensatory mutations, that can partially restore the fitness cost incurred by making the first mutation that evaded the immune response. And now you've got a virus that's almost as fit as before, and it has evaded the immune response. So many properties of an evolving viral population can be described adequately by assuming that each residue in its proteins evolves independently of others. But if you truly want to define the mutational vulnerabilities of a virus like HIV, you do have to define the collective compensatory pathways that HIV can use to evade immune responses, because you want to avoid targeting these regions with a vaccine-induced immune response. And you want to define the combinations of mutations that the virus cannot make and still be replicatively fit, because those are the regions that you want a vaccine-induced immune response to target. That is, in short, for rational immunogen design, you need to determine the fitness landscape of the virus. To illustrate what I mean by a fitness landscape on a two-dimensional sheet of paper, consider a virus that's made up of one protein with two residues. Otherwise, I can't draw it on a two-dimensional plane. So on this axis, each grid point is a mutant amino acid that could arise in residue 1. And on this axis, each grid point is a mutant amino acid that could arise in residue 2. So any point in this two-dimensional plane is a mutant strain of this cartoon virus. And on this axis is shown information about its replicative fitness. So you can see how knowledge of the fitness as a function of sequence will trace out hills and valleys, but of course in a much more high-dimensional space than I've illustrated here. Now, knowledge of this fitness landscape can be really useful for immunogen design, because with a vaccine-induced immune response, you want to push the virus away from one of these hills, force it to make combinations of mutations that correspond to one of these valleys, and you also want to block these mountain passes that correspond to the compensatory pathways that HIV uses to traverse from this hill to another by mutation when this one is under immune attack. So this is the reason why we embarked on a program to try and translate data on the thousands of sequences of HIV proteins that have been collected over the last 30 years to knowledge of this sort of fitness landscape. So our early work began with some very qualitative considerations. And since it was published quite a while ago, I'm going to just tell you some salient features of these old results to set the stage for what I really want to talk about. Sort of a long introduction, if you like. And this early work was led by Vincent Dyrell, a postdoc at MIT in our lab, who is now an independent faculty member in his native Paris, and Karthik Shekhar, who until recently was a graduate student in my lab, but is now postdoc in Aviv Regeev's lab at the Broad Institute. And much of the work in the early phase that I will describe is a collaboration with Bruce Walker, who is a clinician scientist at the Mass General Hospital. So in this early work, we started by saying, instead of thinking of HIV proteins as made up of individual residues, can we partition the HIV proteome into groups of amino acids, with each group being defined such that residues within a group co-evolve, in that combinations of mutations in residues within a group collectively influence viral fitness. But residues in different groups evolve essentially independently of each other. The idea was that if such a partitioning was possible, then one could hope that in some group, or groups compared to other groups, a greater proportion of the combinations of mutations would be deleterious for the virus. Such a group of residues would be immunologically vulnerable to multiple points of T-cell attack, because that would promote the emergence of multiple mutations to evade the T-cell response. But that should likely make the virus unfit because combinations of mutations tend to be deleterious in this group. And notice how this effect would be unlikely to be compensated by mutations in residues in other groups, because by construction, residues in different groups evolve independently of each other. But is such a partitioning possible? So again, without details, some years ago, we used methods that were originally developed in the context of high-energy physics by Eugene Wigner and has been used in many other contexts since then, to analyze these sequences for a particular polyprotein of HIV called GAG, which has 500 residues. And in that polyprotein, we found that there were 5 such co-evolving groups of residues. We determined by mathematical analysis that 2 of these groups were especially vulnerable to combinations of mutations. I should point out that for these 2 groups, we were also able to rationalize why combinations of mutations would be deleterious there from structural data. But I again emphasize that the structure, if you had looked at the structures of individual proteins, you would not have been able to tell that, because these functional reasons came from the importance of assembling multi-protein structures that are critical for viral function. And in many cases, you simply don't have this sort of information to even rationalize your studies with. Now, there are cohorts of patients, I think Bruce was one of the first people to identify them, who are called elite controllers. They are called elite controllers because their T-cell response controls the virus to very low levels. They do not eradicate the virus, but keeps them to such low levels that they don't progress to disease and have very low likelihood of infecting others. So now when Bruce went back and interrogated the T-cell responses in his cohort of elite controller patients, he found that in GAG, his elite controller patients were predominantly targeting these 2 vulnerable groups that we had defined. But that led us to think and try to test these ideas more deeply against clinical information. Our analysis suggested that the 2 groups of vulnerable residues were equivalent in that single mutations in both those groups should be just as likely. But our analysis also suggested that multiple mutations should be more constrained in one group of residues compared to the other. So the prediction was that if Bruce went and took virus samples out of his elite controller patients and looked at the GAG sequences after sequencing them, he should find that in these 2 groups of residues, single mutations are just as likely, but multiple mutations occurred much more rarely in one group compared to the other. So these are data from 103 elite controller patients. This is the number of unique strains of HIV with 1, 2, 3, 4, and 5 mutations, parsed according to the second most vulnerable group in blue and the most vulnerable group in red. And you can see that single mutations are almost as likely, but higher order mutations are much rarer in the most vulnerable group. So taken together, these ideas started to suggest that if you could target multiple points in such multidimensionally constrained groups of residues, you might corner the virus between a rock, namely being killed by the immune pressure, or a hard place, that is, you make mutations that cause you to be replicatively unfit. Now, as you all know, T cells see antigen through their T cell receptor by binding to peptide MHC complexes. There is an enormous diversity of MHC genes in the population. Cohorts of elite controllers are known to be statistically enriched in people who possess certain MHC genes and molecules. The names of those genes and molecules are over there. Over the years, we've spent quite a bit of time thinking about why this may be so. And a dominant reason is that they present peptides, these MHCs present peptides dominantly from regions that tend to be vulnerable by the kind of metric I defined to you. That is, multiple mutations are less likely. But what about people who do not possess these elite controller-associated MHC molecules? Well, if you go and look in the immune databases from clinical studies, you will find that diverse MHCs present peptides from these regions that we have been identifying. But they do so subdominantly. That is, given the whole proteome, they present things from other parts more efficiently. So this suggested to us that for a bug like HIV, for a T-cell-directed response, the immunogen should not be whole proteome, but should be chunks of residues that simultaneously satisfy three criteria. One is they maximize regions that are vulnerable to multiple mutations. Two, they minimize regions that contain these compensatory pathways. And three, they maximize residues that are either subdominantly or dominantly presented by MHC genes that provide broad coverage to a target population. The idea, of course, if that's the immunogen, the memory response that will be generated by vaccination should be specific for these vulnerable bits because the other parts are not there. Now, during natural infection, of course, you get the whole bit, but since the memory response lies ready and waiting, it might be able to target the virus in the vulnerable bits and control it to low levels, much like elite controllers do. So as we started to design these immunogens, we ran into some difficulties. One such difficulty, and the most important one, is that this qualitative business we had done before is insufficient because it doesn't tell us which precise residues in these vulnerable groups to target. Moreover, these vulnerable regions also contain escape pathways. There are just fewer of them. How do we block them? Answering these questions with precision requires that we have a knowledge of a quantitative ranking of the fitness of diverse mutant HIV strains bearing multiple simultaneous mutations. Only then can we know where should we target the immune response such that mutations in those residues will lead to unfit virus in the background of all other mutations that HIV could acquire along the way. So that's what Andy Ferguson, when he was postdoc in our group, started. He's now an independent faculty member at the University of Illinois. And I'm going to start by telling you some of Andy's early work, which was published, and then go on to tell you some more newer things. So the sequence database is just sequences of HIV proteins. Each sequence can be considered to be a vector. Let me call it Z. It's a vector because it's just a list of amino acids. And the sequence database contains information on the probability of observing any sequence. It also contains information on the probability of observing single mutations at every residue, the probability of observing double mutations at every pair of residues, triples at every triplet of residues, and so forth. So any mathematical model that recapitulates these mutational probabilities is a legitimate model for P of Z. So to try to go after P of Z then, we took a page out of work that actually Bill Bialek had done in the context of neuroscience and asked, what is the least biased model for P of Z that by construction fits the 1 and 2 site mutational probabilities that's observed in the data? So how would you do that? I'm going to take you a little bit through the arithmetic of this. So any sequence is a vector zk, and for simplicity, so that I can just illustrate it, let me consider that at each residue, the amino acid is in either wild type or one particular variant called or a mutant. In statistical physics, this would be called an Ising model. In reality, what we do is not this. We have multiple amino acids, but then it becomes a generalization to something that statistical physicists would call a Potts model. But in keeping with this, then, what least biased model mathematically has a meaning. It means it's the maximum entropy model. What do I mean by that? What we are seeking is the probability of observing sequences. This probability distribution has an entropy, p log p, summed over all sequences. We want to choose a p that maximizes this entropy because it minimizes the information we've stuck into it ourselves. And we want to do that maximization subject to the constraint that this probability distribution is normalized and that the one-point mutational probabilities that it predicts is what is observed, and that the pair mutational probabilities that it predicts is what is observed. And you enforce those constraints through something that you learned in mathematics called Lagrange multipliers. And then 2 lines of algebra would show you that the probability of observing strains follows this form, which anybody who had an elementary statistical mechanics class will recognize, something that Boltzmann taught us, with this Hamiltonian having this form, where these numbers, hi and jij's, are extracted by fitting the 1 and 2 point mutational probabilities. Interestingly, because these jij's can be both positive and negative, this kind of structure has a form well known in statistical mechanics in the context of spin glass physics, and also very similar to neural networks that Hopfield defined a long time ago. There are advantages to this. The advantage is now you can take all the machinery developed for that and use it to think about this class of problems. The other advantage is a very personal one. Before my days in immunology 15 years ago, I used to play with these kinds of spin glass models a lot, which is one that I knew quite a bit about them. And the other is I'm now 53 years old, and so memories of your youth are always very pleasant. So I really enjoy working with these things. In any case, the model that you have is not a model for fitness. It's a model for prevalence. Okay? It says what kind of strains are circulating. And you can say that the more fit strains are also likely to be the more prevalent strains. But this connection could be quite complicated. Because the sequences that we have are not viruses growing in petri dishes. They are extracted from individual patients where there has been a massive non-equilibrium battle between that person's immune system and the virus, which has forced certain mutations. So with my physics colleague Mehran Kardar and the ubiquitous Karthik Shekhar, we have thought about the connection between prevalence and fitness in this context. What I'm going to do today is not take you through how we reached that conclusion, but tell you some physical or biological reasons why we find that the rank order of fitness and rank order of prevalence is the same for most circumstances. And that's all we care about, the rank order, which is fitter than the other. The reason is this. In this context, HIV is simpler than some other viruses like the flu. So 3 reasons why this is a relatively simple relationship. HIV has never been subjected, to our knowledge, to an efficient, effective vaccine or natural immune response that has cleared it. So it has not evolved in any particular directed way. 2, there's a humongous diversity of MHC genes in the population. That means that any given region of HIV proteins has been targeted by very few people. 3, if I target the virus in certain regions and force mutations there, if I transmit to someone else, those mutations tend to revert if they are no longer targeted. If you think about these 3 reasons, you will perhaps intuit why it is possible that while the immune response is very important in every single individual for the virus, at the level of the population of viruses, it is a perturbative effect, which is why the relationship between prevalence and fitness can be relatively simple. This is not the case for viruses that have been subjected to effective immune responses. So let me skip the mathematics underlying this, but it's interesting statistical physics, but not important for our discussion today. As you might imagine, my collaborator, Bruce Walker, was not at all impressed by these hieroglyphics and ancient Greek symbols. He asked, well, show me the money. And the money is that you should be able to make predictions of the fitness of viral strains, that I can go make those viral strains and measure them in vitro. So the metric of fitness for us is a quantity called E. So if you give me a mutant strain, we will return a value of E to you. And the theory would predict that the value of E for a mutant minus E for the wild type should correlate negatively with the fitness of that mutant strain divided by the fitness of wild type plotted on these logarithmic coordinates. So we made predictions. What I'll show you today are predictions for 35 different mutant strains. Bruce and his collaborators then made these mutant strains and measured their in vitro replicative fitness to infect human cells. So here's the prediction, and here's the data. 26 data points are shown here, because for the other 9, we had said that the fitness energy was so large that the fitness should be close to 0, and they did not grow out. So we are not perfect, as you can see from this. But with high statistical probability, we can capture about 80% or 85% of the data, depending on the particular system we test against. Briefly, let me mention that we have also been testing this against other kinds of clinical data compared to what I showed you before. And this is work that current postdoc John Barton has been doing, and it's collaborative work with Andrew McMichael's lab at Oxford University. So briefly, what Andrew has is he has sequenced viral strains in individual patients as a function of time. And he has also determined what the T-cell response is that they are putting. In these patients, the virus escapes. It makes mutations that evades the immune response. So the first question that he asked is, can you tell me that of the various possible mutations that could occur for escape, where do the mutations actually occur in these individual patients? Using our fitness landscape-based ideas, and without details, at least for this cohort of patients, we have pretty high accuracy in predicting where the mutation will occur. The only thing I will say about that work is our results highlight the importance of these collective compensatory pathways. So if you target the same epitope in 2 different patients, escape can occur in widely different times, for example, because it matters very much what the sequence background is that the patient inherited in other parts. And our approach incorporates those things, doesn't look at it sequence residue by residue. He then asked us a harder question: can you rank order the times at which escape in these individual epitopes actually occurred? That this will happen first, second, and so forth. So for over a century, ever since Sewall Wright and Fisher, people have done evolutionary dynamics. But to my knowledge, mostly with toy fitness landscapes. So what we did, well, we have a fitness landscape of a real virus, we think. So let's do Wright-Fisher evolutionary dynamics with this fitness landscape and see what we can predict. So again, for this cohort of patients, this is our predicted evolutionary generations. That's how you measure time in these Wright-Fisher models of escape. This is the real escape time measured in days. And again, we are not perfect, but you see that we are not bad either. And this data is real human clinical data in individuals. This is important for us because the ability to predict how long control will be maintained with a certain vaccine-induced T-cell response is important for in the therapeutic vaccination setting. Anyway, based on all these things, we have designed an immunogen, doesn't matter what it is, which are chunks of different parts of the proteome, which have been synthesized into one long peptide. And the trouble is that these long peptide immunogens are often not very immunogenic when delivered in the usual ways. A cancer vaccination has confronted this, is trying to confront this challenge, and we are trying to too. When I say we, I use the word we very, very loosely. My colleague Darrell Irvine at MIT has developed a nanoparticle-based approach for such long peptide immunogens in the context of cancer vaccines. And he has published a paper in one of the tabloids where he shows that, at least in mice, in this cancer setting, you get a large CTL response. So this, our immunogen has been incorporated here, and he's collaborating with Dan Baruch at Harvard Medical School and trying to optimize this delivery methods in monkeys, We are also following other approaches like adenovirus vector-based approaches and so forth. And if such an optimal delivery method could be defined, then what Bruce wants to do is put this into humans who are currently infected and see if their immune response or T-cell response can be redirected such that they target these vulnerable regions, control the virus to low levels like elite controllers, and then take them off of ARV treatment and see if they can control. This may all be pipe dreams, okay? There may be some hope, however, because these people from EPFL published a paper in Lancet earlier this year where they took our 3-year-old study where we had just these qualitative bits and they mixed it with something called GMCSF, which is people with much higher pay grade than me say that is a lousy adjuvant, but is FDA approved. And they did this experiment that Bruce wanted to do with a small group of people. They did not convert them to elite controllers, but they got drop in their viral loads. So perhaps if done with the better immunogen, with better delivery methods, there may be some hope. I want to use the rest of my time to switch and tell you a bit about the antibody side of this problem as well. This is work that has been led by postdoc Xinshen Wang in my group, and it's a multidisciplinary collaboration between me, my theoretical physicist Mehran Kardar, Dennis Burton, who plays a major role in the isolation and characterization of broadly neutralizing antibodies and so forth, and Dane Wittrup, who is a bioengineer at MIT. And this work will also appear next month in another tabloid. So everybody knows that high affinity antibodies are produced by a process called affinity maturation. Just for completeness, let me briefly remind you of how this Darwinian evolution process occurs in a short time. So naive B cells, when they get activated by a certain antigen, they can seed germinal centers. This germinal center is a structure in your lymph node where there's a region called the dark zone where B cells, they start to multiply, but a gene called AID gets turned on, which introduces mutations into the B cell receptor, which is what binds to antigen. These mutated B cells then go to some region called the light zone, where follicular dendritic cells display the antigen on their surface. And these B cells, if they can bind to this antigen, they will internalize these antigens and display peptide MHCs derived from the antigen's proteome. And then they undergo a Darwinian evolutionary competition between each other to seek limited amounts of interaction with T helper cells, which then, if they survive, and if they don't manage to win this competition, they die. If they survive, some fraction of them become plasma cells which secrete antibodies, another fraction become memory cells, and a large fraction is recycled and go through rounds of mutation and selection. This problem of affinity maturation has been studied greatly. Ever since Hermann Eisen, my dear and beloved colleague who passed away 3 months ago, did the key experiments in 1964 to show that affinity maturation actually happened, and several people have made very important contributions experimentally. This is also a problem that has been extensively studied by computation. In fact, Tom Kepler has done some of the most seminal work in the field of understanding how affinity maturation occurs through computation. And therefore, I thought that I would never work in this problem. It's a closed problem. It's what I always believed. Well, what made me change my mind? Well, the induction of broadly neutralizing antibodies will likely require immunization with more than one variant antigen strain, because if you have just one variant antigen, one antigen, this Darwinian evolutionary process will produce high affinity antibodies that are strain specific. So it seems likely you will have to immunize with multiple antigens. That raises the question of which antigens. What should be these variant antigens? Many people are working on this question. But it also has other questions that arise. What should be the temporal pattern in which you administer these variant antigens? Should they be cocktails? Should they be a sequence? Should they be other schemes? What should be the concentrations of these antigens? And I go to these meetings where I used to keep hearing people talk about these questions. I always wondered that this beautiful work done in the past on affinity maturation why don't you use that as a guide to answer these questions? Because the space of possibilities from which answers to these questions are drawn is huge. And so it seems unlikely that by experimental sampling alone, you will comprehensively sample this whole region and get the golden nuggets. But the trouble is that affinity maturation in the presence of multiple variant antigens is not very well understood. Because most studies in the past, including most of Tom's beautiful studies of the past, were done with single antigens. So that's why we have developed an in silico model for affinity maturation in the presence of multiple variant antigens with two goals: to focus experiments on a few promising choices, and the other is to obtain a more fundamental understanding of affinity maturation in this more complex setting. And what I'll do today in the 5 or 6 minutes, maybe I'll take 7 minutes or so to tell you is that I'll just address the question of the effect of different temporal patterns in which these variant antigens are administered. Let me skip the details of the model, okay, and just focus on the results. And if there's interest in the question-answer period, I will tell you some details about the model. Because of the interest of time, I'll skip the details of the model. So we have studied three variant antigens using this computational algorithm. One that we call wild-type antigen. And then there are two mutant strains with non-overlapping mutated variable residues that surround the... So any broadly neutralizing antibody that you'll have will have some conserved residues in their epitope, otherwise not going to be broadly neutralizing. But there are variable residues all around, there are glycan shields all around, and that is that. So when I say mutations, I mean mutations in the variable residues that surround this conserved set of residues. Is that okay? And we've thought about different extents to which these strains are separated in terms of the number of mutations. But I'll focus only on one case in this short talk, in the short time available. We've studied 3 different immunization schemes in silico: a cocktail, then you give one, then you prime with one, and then you boost with a cocktail of the others, and you do sequential immunization with the various variants. Now, for each scenario, we look at the antibodies that emerge from our calculations, and we screen them for their affinity against a panel of viral strains that Mike Seaman has developed that is used frequently to test for breadth of coverage. And the breadth of coverage is defined as the fraction of strains in this panel that bind with an affinity above a threshold. We run the calculation for each setting many, many times. These are stochastic evolutionary trajectories. We run the calculation many, many times and collect statistics of the breadth of antibodies that are produced. And so we get an answer of the probability of producing antibodies with different breadths in any individual that is vaccinated. And the results are presented as histograms. So this is what happens when you immunize with a cocktail of the variant antigens. This is the probability of getting breadths of different coverage, one being complete coverage. And you see in this case, you do not get cross-reactive antibodies with any measurable probability. We also find that most of the germinal centers just collapse. They don't produce antibodies at all. And so antibodies are also produced at low titers. To see why this is the case in this setting, consider the case where when B cells interact with follicular dendritic cells, even though there are variant antigens, they see only one antigen at a time. For this case, it makes it very clear why this happens. In early stages of affinity maturation, the B cell receptors have not developed strong interactions with the antigen. So they certainly have not developed strong interactions with the conserved residues, which is shown in red here. So imagine a B cell that gets selected during one round of mutation selection by a particular variant, mutates, goes back and now encounters a different antigen, the variant antigen. Because it doesn't have strong residues with the interactions with the conserved residues at this point, the chance that it will be cross-reactive to this is small. And so the most likely outcome is that it will apoptose. It will not succeed in selection. That is to say, these conflicting selection forces imposed by the variant antigens can frustrate this evolutionary process of affinity maturation in this setting. Actually, the problem we have been handed is a very hard one. It poses some basic question in evolutionary biology. See, at each point in time during this affinity maturation evolutionary process, the fitness that's being maximized is affinity for whatever it's interacting with. We are asking, how do you organize the variant antigens spatially and in time such that a quantity different from this local fitness, namely the breadth, is optimized? For example, if you now consider the case where every time the B cell receptor B cell comes and is selected, it sees all kinds of antigens simultaneously in the synapse. You know what's going to happen? You're going to get strain-specific antibodies, because as long as you can see one of them repeatedly every time, you'll be fine. Won't give you breadth. You get higher antibody titers. Let me just show you two other cases. One is if you prime with one variant and boost with a cocktail. Now you see, with some low probability, you do see these cross-reactive antibodies. You also see them in moderate titers. The reason is that while this frustration still happens when you boost with the cocktail, it's partially alleviated because in the first prime, you develop some strong interactions with the conserved residues. We think this may be what is happening in natural infection, where you get one variant and then it diversifies. And so you get broadly neutralizing antibodies with low probability. And what do you see in people? It happens in very few people. And also, after 2 or 3 years, because low probability events are rare and take time to happen. In this setting, we have found some special circumstances where you can get cross-reactive antibodies with high probability. And this is consistent also with the beautiful work that Bart Haynes is doing. Tom is involved in some of them, where you can trace back the lineage of the broadly neutralizing antibody and the virus that's competing with it. You see these very special circumstances when this thing emerges. We see those types of things too in this setting. But our naive view is that it may be very difficult to replicate these very special circumstances in every patient. And even if you were able to do that, because evolution is a stochastic process, it may be hard to replicate this exactly in every individual. So perhaps mimicking successes during natural infection, which are so important to understand, may not be the most fruitful vaccination strategy. Now here's what happens when you immunize with the variant antigens. Now you see broadly neutralizing antibodies occur with much higher probability. You also get them in high titers. And the reason, at least in our simulation, this is the following. This temporal separation of conflicting selection forces, and its impact can be best seen by looking at the footprint of the antibodies on the antigen as you go through this process. When you prime with one, you get a broad footprint of the antibody on the conserved residues and the variable residues of this particular antigen. You come with a second variant, and we see that two kinds of lineages develop. In one, because there are strong interactions with the conserved residues, you just remove interactions with the newly mutated sites, and you increase those with the conserved residues. In another, you generate strong interactions with the newly mutated residues. Now when you introduce the third variant, which has non-overlapping mutations, this class of trajectories or antibodies, these lineages, have a hard time surviving because they have to do a bunch of stuff. They have to undo all these interactions and build new ones. Whereas this one, they just take off a few more interactions and focus down on the conserved residues. So since time is short, let me not belabor the fact that we have tested these in model experiments with mice. So what has been done is 4 variant antigens, which are monomeric GP120, so they can never give you neutralization. You can only test the principle that they will focus down the conserved residues, because the real virus has a trimer on the surface. And these were then, mice were immunized as a cocktail and sequentially. And the question that was asked is, which case focuses down on the conserved residues? And let me quickly show you the data. This is for a cocktail. These are the 4 variants, and you see the antibodies have a broad distribution of activity to them. This is sequential. You get a much narrower band of focused responses. And finally, what they did also was they took this antigen, bound it to VRCO1, which is a great antibody focused on the CD4 binding site, put serum from mice, cocktail, and sequential, and asked, does the serum bind? If it binds, that means the epitope is not this. There are other epitopes. If it doesn't bind, the epitope is focused on the conserved residues. And this is what you see amongst percent cells that bind. Sequential, very few. Cocktail, quite a few. They have then also taken sequentially immunized mice and extracted monoclonal antibodies from them, and done exactly the same experiments with 2 monoclonal antibodies, and they show the same feature. So while getting broadly neutralizing antibodies is a very, very hard problem, we have touched on just a bit of it. And the bit of it we have touched on suggests that if the right antigens were available, sequential immunization might offer a better chance of getting cross-reactive broadly neutralizing antibodies than cocktails and other variants of immunization schemes. So with that, I'll stop. I think I've thanked all the people along the way, and I apologize that it took 5 minutes longer than planned. Thank you.